Welcome everyone to a new episode of Biology. Today we'll talk about regulation of the cell cycle. The key concept is the cell cycle regulation is necessary for healthy growth. The main ideas: number one, internal and external factors regulate cell division. Number two, cell division is uncontrolled in cancer, and we will study the cancer cells. The first step, internal and external factors regulate cell division. Both internal and external factors regulate the cell cycle in eukaryotic cells. And keep in your mind that the eukaryotic cells are the cells that have nucleus and membrane-bound organelles. External factors come from outside of the cell. External factors include the messages from nearby cells or from distant parts of the organism's body that are the external factors. Internal factors come from the inside of the cell itself and include several types of molecules found in the cytoplasm. Both types of factors work together to help your body control the process of the cell division. What about the external factors? The external factors that help regulate the cell cycle include physical and chemical signals that come from outside of the cell. One example of a physical signal is cell-to-cell contact, the touch between the two cells. Most mammal cells grown in the laboratory form a single layer on the bottom of a culture dish. So the mammal cells grown in the laboratory in a culture dish, they form a single layer. Why? Once a cell touches the other cells, it stops dividing. Once a cell touches the other cells, it stops dividing. So the cell-to-cell contact, this is a physical external factor that controls the cell cycle or the cell division. The exact reason for this phenomenon is unknown. One hypothesis is that receptors on the neighboring cells bind to each other and cause the cell's cytoskeletons. Do you remember the cytoskeletons? To form the structures that may block the signals that trigger growth. Again, one hypothesis is why when the cell touches the other cell, it stops dividing, is that the receptors on the neighboring cells bind to each other and cause the cells, the cytoskeletons, to form structures that may block the signals that trigger growth. Many cells also release chemical signals that tell other cells to grow. These are chemical external factors. For example, growth factors are a broad group of proteins that stimulate cell division. Growth factors bind to receptors that activate specific genes to trigger the cell growth. So the growth factors, these are chemical external factors, which are a group of proteins that stimulate the cell division. So the growth factors bind to receptors in the cell that activate specific genes to start the cell growth. In general, cells grow and divide in response to a combination of different growth factors, not just one. A normal animal cells at the top respond to external factors and stop dividing when they touch each other, as this one in the culture dish. They are normal cell. This is a normal cell growth because they form only a single layer. When the cell touches other cells, it stops dividing. But in cancer cells, it fails to respond and form clumps. Clumps are like chunks or a group of cells. This is the cancerous cell growth. Okay, some growth factors affect many different types of cells. For example, platelets are sticky fragments of bone marrow cells. The platelets, this is part or a main component in our blood, which are sticky fragments of bone marrow cells. Platelets form clots that help stop bleeding. Yes, when someone is cut, so the platelets is responsible for forming clots to stop the bleeding. Platelets store a type of growth factor that helps your body repair wounds by triggering the growth of many different cells. So it have a special type of growth factor that help our body repair the wounds by triggering the growth of many different cells. Other growth factors have more specific targets, like what? For instance, erythropoietin. Erythropoietin stimulates the production only of cells that will become red blood cells. So erythropoietin is a growth factor which is more specific or has more specific target. Like erythropoietin stimulates the production only of cells that will become red blood cells. The red blood cells carry the oxygen. Yes, we already know this. If you moved from the coast to the mountains, your blood oxygen levels would be lower because the air pressure is lower at higher altitudes. So at the higher altitudes, like the mountains, there is lower blood pressure. So in this case, your blood oxygen levels will become lower. The decrease in the blood pressure or in the blood oxygen, the decrease in the blood oxygen levels would cause your body to produce more erythropoietin, to produce more red blood cells, to get more oxygen that our body needs. That factor would increase the number of red blood cells and raise your blood oxygen levels. Various hormones may also stimulate the growth of certain cell types. In particular, growth hormones results in bone growth, also affects your protein and fat metabolism. So the various hormones may also stimulate the growth of certain cell types. In particular, growth hormones result in bone growth and also affect your protein and fat metabolism. What about the internal factors that control the cell cycle? When external factors bind to their receptors, they can trigger internal factors that affect the cell cycle. Two of the most important and well-studied internal factors involved in the eukaryotic cell cycle are kinase and cyclins. Kinase and cyclins. A kinase is an enzyme that, when activated, transfers a phosphate group from one molecule to a specific target molecule. So kinase and cyclins, these are the two internal factors that are triggered when the external factors bind to the cell receptors. So two of the most important and well-studied internal factors involved in the eukaryotic cell cycle are kinase and cyclins. A kinase is an enzyme. This is important. A kinase is an enzyme. When activated, it transfers a phosphate group from one molecule to a specific target molecule. This action typically increases the energy of the target molecule, or it changes its shape. Increase the energy. Do you remember the ATP and the ADP? When we added the phosphate group, so we increase the energy. Your cells, your cells have many types of kinases, and they are almost always present in the cell. Those kinases that help control the cell cycle are activated by cyclins. This is the second internal factor, cyclins. So kinases help control the cell cycle and are activated by cyclins. So cyclins activate kinases. So what about cyclins? Cyclins are a group of proteins that are rapidly made and destroyed at certain points in the cell cycle. Cyclins are a group of proteins that are rapidly made and destroyed at certain points in the cell cycle. So the cyclins rapidly made and destroyed, while kinases, they are found all the time in the cell. Cyclins activate kinases. These two factors help a cell advance to different stages of the cell cycle when cells bind to each other. Then we move to another part, which is apoptosis. Apoptosis. So what's apoptosis? Just as some cells need to grow and divide, other cells need to die, and this is important. Why? Let's see. Apoptosis itself, it means programmed cell death. Programmed cell death. Apoptosis is programmed cell death. Why? It occurs when internal or external signals activate genes that help produce self-destructive enzymes. So this is a self-destructive enzymes that are activated inside the cell to start apoptosis. Many questions remain about this process, but let's see. What is known is that the nucleus of an apoptotic cell, tends to shrink and break apart, and the cell is recognized by specialized cells in the immune system. So the nucleus of apoptotic cell started to shrink and break apart. Then the cell itself is recognized by specialized cells in the immune system. These cells very tidily or organized gobble up or eat the apoptotic cell and recycle its chemical parts for use in building other molecules. So, apoptosis is programmed cell death for some cells. The apoptotic cell, its nucleus started to shrink and break apart. Then, a specialized type of cells in the immune system recognize this apoptotic cell. Then, these cells very tidily or organized gobble up the apoptotic cells, the specialized cells of the immune system, gobble up or eat the apoptotic cell. Then it recycles its chemical parts for use in building other molecules. Okay, a classic example of apoptosis is human embryos. Yes, look at the picture to the left. This is a human embryo in the early stage inside its mother's body. Can you see the toes and fingers? These fingers, they are called webbed digits because there's like a web between each two digits or two fingers. So the human embryos have webbed digits early in their development. Then the cells between the digits, the cells between the fingers undergo apoptosis, and it will undergo apoptosis during the later stages of development. Then we have unwebbed fingers and toes. As a result, the baby is born, as you can see, as all of us, with unwebbed fingers and toes. The second main idea in our lesson today is the cell division is uncontrolled in cancer. So we will talk about cancer. Cancer is the common name for a class of diseases characterized by uncontrolled cell division. Cancer is uncontrolled cell division, or cancer is the common name for a class of diseases characterized by uncontrolled cell division. Cancer arises when regulation of the cell cycle breaks down. Cancer arises when the regulation of the cell cycle breaks down. Unlike healthy cells, cancer cells grow in a culture dish to continue to divide, even when surrounded by neighboring cells, as we already saw in the culture dish. If you remember, when it formed clumps, yes, it continued to grow. Okay, cancer cells can also continue to divide in the absence of many growth factors required for division in healthy cells. Can you imagine this? The cancer cells, they continue to divide even in the absence of many growth factors that are required for division in healthy cells. As a result, they divide much more often than do healthy cells. As a result, they divide much more often than do healthy cells. Cancer cells form disorganized clumps or group of cells called tumors. In a benign tumor, the cancer cells typically remain clustered together or gathered in masses or chunks. This is in a benign tumor. So what about this means what? This means that the tumor may be relatively harmless and can probably be cured by removing it. So the benign tumor, the cancer cells, typically remain clustered together or gathered in masses or chunks. This means that the tumor may be relatively harmless and they can probably be cured by removing it. But, however, if a tumor is malignant tumors, some of the cancer cells can break away from the tumor or metastasize. So metastasize is a part of this is a process of a part of a cancer cells started to break apart and move away. This is only in malignant tumors. So these breakaway cells can be carried by the bloodstream or lymphatic system to the other parts of the body where they can form more tumors called metastasis. Once a tumor metastasizes, it is much more difficult to entirely rid the body of tumor. So we cannot cure or remove the tumors from the body when they metastasize, or this is in malignant tumors. As you can see, this is a normal cell tissue, and there is a cancerous cell. This is the bloodstream. After some time, all these or the number of cancerous cells start to increase, then some of it move to the bloodstream. This is in case of malignant tumors. But why are tumors harmful? Cancer cells do not perform the specialized functions needed by the body. The cancer cells do not perform the specialized functions needed by the body. In the lung, for example, cancer cells do not exchange oxygen with carbon dioxide. In the brain, cancer cells do not transmit the carefully ordered electrical messages needed to interrupt information. So the cancer cells do not perform the normal processes of the cell. Therefore, the body has large clumps of rapidly dividing cells that require lots of food and a hearty blood supply, but that contribute nothing to the body's function. In addition, a growing tumor can exert great pressure on the surrounding organs. Yes, for instance, a tumor growing inside the skull. Can you imagine that the skull is like a closed box and there is a tumor inside? So it will cramp the brain for space, and some regions will be unable to function properly. Yes, because this is overcrowded. If cancer cells continue to grow unchecked, they will eventually kill the organism for sure. Cancer cells come from the normal cells that have suffered damage to the genes that help make proteins involved in the cell cycle regulation. So cancer cells come from the normal cells that suffer the damage to the genes that help make proteins, which are involved in the cell cycle regulation. Most cancer cells carry mutations or errors in two types of genes. So what are these two types of genes that have mutations in cancer cells? One type cell called oncogenes. Oncogenes accelerate the cell cycle. Do you remember when we said that uh, cancer is the uncontrolled cell division? This is because the cancer cells carry mutations or errors in two types of genes. One mutation a mutation in one gene called oncogenes, which accelerate the cell cycle. This is the uncontrolled cell division. The second type acts as cell cycle breaks. This one which stops the cell cycle. So the mutations in these genes can be inherited, yes. For instance, some breast cancers appear to be caused by inherited errors in specific genes. Other mutations can be caused by exposure to radiation or chemicals. These are the two causes of cancer, or the mutations that cause cancer. Some of it are inherited, others can be caused by mutations or exposure to radiation or chemicals. For example, some skin cancer are due to DNA damage caused by ultraviolet radiation from the sunlight, as this one, as the moles. So this cancerous mole means the birthmark or the beauty sign. So the cancerous mole is an example of a skin cancer, which may metastasize quickly, which may move in the bloodstream to affect another part. These are examples of cancerous moles. Substances known to produce or promote the development of cancer are called carcinogens. Carcinogens. So these are the substances can promote or produce cancer are called carcinogens. Carcinogens include tobacco smoke and certain air pollutants, which are both associated with lung cancer. Carcinogens include tobacco smoke, and this is for the cigarettes, and certain air pollutants, which are both associated with lung cancer. Some mutated forms of oncogenes are even carried by viruses. One such virus can cause cervical cancer. Some mutated forms of oncogenes are even carried by viruses. One such virus, for example, can cause cervical cancer. Standard cancer treatment often involves both radiation and chemotherapy. Here we came to the treatment of cancer. It involves both radiation and chemotherapy. Radiation therapy is the use of radiation to kill cancer cells and shrink tumors. Radiation therapy works by damaging a cell's DNA so much that the cell cannot divide because the DNA is damaged enough to stop the cell from dividing. Radiation usually is localized. Radiation usually is localized, that is, it is used Its use is targeted to a specific region because it can also hurt healthy cells. This is radiation. What about chemotherapy? Chemotherapy uses certain drugs, often in combination, to kill actively dividing cells. But like radiation, chemotherapy kills both cancerous and healthy cells. However, chemotherapy is systemic. These are drugs that travel throughout the entire body. Medical researchers use laboratory grown cancer cells in their search for cancer treatments. Much of what is known about the cell cycle has come from studies that use cancer cells. Yes, the most famous cancer cells used for research are called HeLa cells. HeLa cells were originally obtained in 1955 from a cervical tumor removed from a woman named Henrietta Lacks. And this is the end of our session for today. Hope you understood the lesson. See you in the next episode and goodbye.